The overall goal of this procedure is to successfully transform foreign DNA and knock down gene function in Pristioncus pacificus nematodes. For transgenesis, start by preparing the proper genomic DNA with compatible cohesive ends. For RNA interference, synthesize the gene of interest using a double-stranded RNA transcription kit. Then, microinject the Pristioncus pacificus gonads with a DNA or double-stranded RNA. Screen the F1 population for the selected marker phenotype in transgenesis and selected for the knockdown phenotype for RNA interference. For transgenesis, continue to monitor results of F2 transgenic worms to obtain results that show specific gene expressions using GFP reporter through fluorescence microscopy.
Hi, I'm Ray Hong, California State University, Northridge, and this demonstration will outline the steps for genetic manipulation in Pristioncus pacificus nematodes, an emerging model organism. Performing RNAi and transgenesis can answer key questions in developmental biology and behavior by characterizing gene functions and expressions.
Individuals new to this method will benefit from viewing how to place the worm on the injection pad and to inject the worm on the correct focal plane where the gonads are visible. Unlike C. elegans gonads, the Pristioncus pacificus gonads cross each other dorsally and migrate ventrally as the nematode develops from late J4 into adults. In addition, genomic DNA digested with compatible restriction enzymes should be added to your transgenes and co-injection marker to make your injection mix.
Jessica Single-Portman, a lab technician in my lab, will demonstrate microinjection in Pristioncus pacificus.
For transgenesis, 3 DNA constructs are required for injection: the dominant co-injection marker, the target reporter transgene, and genomic DNA. DNA constructs are digested with compatible restriction enzymes. The DNA constructs are collected using an ethanol precipitation protocol. The injection mixture is made to the concentrations listed in the table. Verify the pressure gauge settings of the nitrogen tank. Now load 1 to 2 microliters of injection mixture into the needle. Place the needle in the microinjection manipulator. Break the needle tip by touching it slightly to an edge of a thinly pulled capillary tube placed on a slide and set it at an idle position. Now pour paraffin oil to cover the plate of J4 young adult worms. Pick a single worm and transfer it to an injection pad. Under the scope, position the worm's gonad in the direction of the needle, with the vulva away from the needle. Now lower the needle into position of the microscope view. Bring the top gonad and the needle into the same focal plane. Gently poke the worm and pump in the injection mixture. Notice the eggs separating towards the distal tip, indicative of a successful injection. Reposition the needle to inject the lower gonad and repeat the injection. Since this gonad is under the gut, the spreading of the eggs will not be seen, and therefore it's more difficult to gauge the success of the injection. However, post-injection, the worm should remain intact without protruding gonads. Replace the needle back in the idle position. Add a drop of M9 buffer to rescue the worm. Using some OP50 bacteria on a pick, touch the worm with the OP50 such that it sticks. Now transfer the worm onto a plate seeded with 50 microliter spots of OP50. store the injected worms at 20 degrees Celsius for roughly 4 days to lay and grow F1s. Now screen the worms for the selected phenotype. Pick single F1s with the observed phenotype to new seeded plate and store at 25 degrees Celsius. Finally, for transgenic lines, transfer single F2s into individual plates. Store subsequent generations at 20 degrees Celsius. Manipulation of gene function at the organismal level by RNAi and transgenesis are key aspects of the P. pacificus model for evolutionary developmental biology. Compared to a wild-type worm on the left, the stable F4 PPRL3 transgenic line results in a dominant roller that shows head neuron GFP expression. Conversely, this RNAi experiment generates PRL1 mutants that exhibit complete knockdown of the rolling locomotion usually observed in the PRL1 roller gain-of-function mutant TU92.
Once mastered, each injection can be completed in 5 minutes. It's important to remember that all F2s from independent F1 transgenic roll lines need to be singly picked to establish independent F2 progeny. This is because we found GFP expression to be highly variable among F2 lines, and transmission of the roller phenotype does not correlate with the G expression level.